Hi everyone, this is topic 5.6 for chromosomal inheritance. So the big idea for this topic is basically we know genes and alleles, it gets segregated or separated into alleles and gametes during meiosis, and those can get passed on through fertilization to make offspring. And then here we also want to explore those bigger picture genes found on the chromosomes as a whole, and what happens when chromosomes maybe get inherited abnormally and what that leads to. So first, just a quick review of some different ways that sexually reproducing organisms increase genetic variation. First one being law of segregation. So the separation of alleles during gamete formation. Each gamete carries only one allele for each gene, and so each gamete receives only one parental allele. Segregation of parental alleles into gametes provides opportunity for more varied combinations when fertilization occurs. So that's just saying that those alleles separate when gametes form. Right? We have a tetrad, right? Two homologous chromosomes. Each one has a different allele, and each of those will go into one single gamete by the end of meiosis. All right, next up is the independent assortment. So another Gregor-Mendel law or conclusion. And this is the one that we use to test chi-squares to determine if our data is significantly different from our expected outcomes. And we do that by looking at two or more traits. And the idea is that two or more traits should divide or separate independent of each other, meaning those genes are not linked to them and that they're found on different chromosomes. And so we would expect to see normally in our expected outcomes is that two genes go in a 9:3:3:1 ratio when we do a dihybrid cross. 9 out of 16 are dominant for both, 3 out of 16 are dominant for one and recessive for the other, 3 are the reverse of that, recessive for one, dominant for the other, and then 1 out of 16 is recessive for both. And so a big idea is like these genes and the genes we get from our parents are inherited randomly, right? And so there's no preference for one allele or gene over another, and there's usually not linked together by being found on the same chromosomes. And this allows for more possible different gametes or more possible gene combinations when fertilization occurs. Right, so we can look at some different possibilities of a pea plant or pea shape. We have two sets of homologous chromosomes here. So if we wanted to look at like the ploidy level of these, we could say 2n equals 4. Right? And then after meiosis, we could say like n equals 2 here, because we're going from diploid to haploid. But also we have different genes. And so if this was like small a, and this one's capital A, we can see as gametes are made, those alleles assort independently, right? Big A isn't likely to make more big B's next to it, or small a's don't have small b's next to them. It's random. And so that distribution is what we'd expect when we do our Punnett squares and crossings. And then the last way to increase genetic variation, random fertilization. So each time fertilization occurs, a sperm cell and an egg cell fuse together to form that fused egg cell or zygote. Those sperm cells and egg cells fertilize randomly. There's no preference for egg cell, no preference for sperm cell. Basically, whichever one happens first is the one that will create the offspring. Okay, and so when we think about like chromosomal inheritance, we can think sometimes about human genetic disorders. And this has to do with when alleles mutate and get passed on from parents to offspring. So chromosomes can carry mutated alleles. The law of segregation and independent assortment explain how these mutated alleles can be randomly distributed into gametes. And so another big reason that contributes to genetic diversity is mutations. There are at any time like hundreds of mutations that happen between gamete formation as well as fertilization that is completely different from either parent, right? So right off the bat, we are already genetically different, not just 50% from mom and 50% from dad, but the 50% from both of them are going to be slightly different than what they started with because of these random mutations. And so mutations can manifest as genetic disorders when variations negatively affect the function of the offspring. Keep in mind, mutations are random. There's no positive or negative umbrella statement we can make about mutations because they are random and they can provide good things, they can provide neutral things, they can provide bad things. But this is one of the ways that can lead to chromosomal disorders. And these disorders can be caused by a single mutated allele, and we'll take a look at like sickle cell disease as an example of that, or specific chromosomal change dealing with larger parts of the chromosome, right? And so disorders can be attributed to specific chromosomal changes. There are well over 6,000 known genetic disorders, all of those stemming from differences in alleles or chromosomes. And so there's this process that we call non-disjunction, which is all about the failure of chromosomes to fully separate during the formation of gametes. And so non-disjunction will typically happen is an error in either anaphase 1 or anaphase 2. Okay. And so the end result is that we have too many or too few chromosomes in those gametes or sex cells. And if we have too many, that is addition. If we have one less than we need, that's deletion. Right. And it's important to kind of, when we look at the results of non-disjunction, how do we know if it was an error in anaphase 1 or anaphase 2? Well, anaphase 1, if you remember, separates homologous chromosomes. So those big full chromosomes get separated to each side. This is different from anaphase 2, which is all about sister chromatids splitting up. Right, and so if we look at some images here, we have a normal process of meiosis happening. Here we have non-disjunction happening in anaphase 2. And so those sister chromatids, instead of going both ways, now we have one that doesn't have any sister chromatids or 2 chromosomes, that one having one more than needed. Versus non-disjunction in anaphase 1 or meiosis 1, that leads to, we have 2 full cells that are both having 2 more or 1 more chromosome than needed, 2 gametes that have 1 less chromosome than needed. So anaphase 1 will typically have a larger impact on more cells compared to anaphase 2. And so non-disjunction is almost used simultaneously with this chromosomal abnormality, which is called aneuploidy. Abnormal chromosome number due to an extra or missing chromosome. And so what we'll say is that when there's only 1 chromosome at the spot 1 through 23, that's called monosomy. And when there's 3, it's trisomy. And so we use this with our chromosomal number to distinguish what specific genetic disorder our offspring or kids born with. And so we can use charts and data to track genetic disorders, and we look at these visuals called karyotypes. And so a karyotype is just showing a chart of chromosomes of an individual, arranged and numbered by size. So everyone has the same relative chromosome 1. There are the same genes on chromosome 1 for everyone, but that's not to say there aren't mutations that happen. Chromosome 2 has a bunch of different genes on it. And so you can start to get dominant and recessive alleles, and then that's how you know what traits get expressed and which don't. And so what we say is that chromosomes 1 through 22 are called autosomes, or non-sex chromosomes or body chromosomes. They're just this is a normal Punnett square. And then we do chromosome 23, the last pair, are sex chromosomes. And so a person's sex chromosomes can be XX or XY, XX for female, XY for male. Okay. And so we'll just take a quick look at some examples of genetic disorders caused by non-disjunction. First one is called triple X syndrome. This is a some type of error, usually in egg or sperm cells, where reproductive cells have one extra chromosome. And so the offspring will have one extra sex chromosome that we can see here, XXX. The offspring would be a female here, and it could have different issues related to sex-linked traits. So maybe that's like hormone regulation or something else. Next up is probably the most common genetic disorder, trisomy 21, also known as Down syndrome. This is when there is an extra chromosome at the chromosome 21 spot. And so this is the earliest known genetic disorder, and then there are certain characteristics that go along with this: flattened bridge, low muscle tone, mild to moderate low range of IQ. And then we have other different types of disorders too that can impact the alleles or genes. And so these occur at single genetic points. So another example or common example is Huntington's disease. Huntington's is a progressive and eventually fatal neurological disorder. This is basically causing degenerative abilities in the muscles and the brain, causing people to lose abilities to function in different parts of their body. So this is autosomal dominant, so that's just a normal non-sex-linked trait. And then dominant is just dominant, big A or big P, however you want to call it. And this just occurs at a single gene. Okay, so just a single mutation can cause this. The thing about Huntington's disease is that it doesn't start showing symptoms until later in life after you already had kids. And so people who have Huntington's disease might not know it yet and would potentially pass it on to offspring. And so we can do pedigrees or a pedigree analysis to determine what's the likelihood that offspring born from a couple, what's the likelihood that they have Huntington's disease. You could get genetic screenings for something like that too. Next up is Tay-Sachs disease. So this is a partial deletion of chromosome 15 inherited by the parents. Certain genetic disorders people have found are inherited in different populations. And this one, for example, is more common in Eastern Jewish populations. About 1 in 29 are carriers for this Tay-Sachs disease. And so what happens is because of this mutation, there's no enzyme that breaks down fatty substances, which can build up to toxic levels in the brain. So this can lead to muscle twitches, nerve damage, involuntary muscle movement, and eventually death. And so Tay-Sachs disease, we can see a simple mutation, again, at that chromosome 15 spot, impacts an enzyme or lack of enzyme being present, which can cause things to accumulate, fatty acids to accumulate in nerve cells or neurons. Okay, so if we just look at this example problem real quick, we can see what is the most accurate prediction of the inheritance pattern illustrated in the karyotype. You want to look for any abnormalities. If there are none, then we would just say this is a normal individual with no apparent chromosomal disorders. Here, however, we do see there is 3 chromosomes at the sex chromosome spot. And so if you look at the options real quick, you can kind of see that what we have here is that one chromosome failed to separate. So not necessarily alleles. Alleles would be found on whole chromosomes, so that's too small of an answer. Too many gametes, no, that would have 3 chromosomes at each spot, which we don't see. Chromosomes failed to separate during gamete formation. I would star that one. That sounds good. Chromosome was lost during gamete formation. That would cause one less chromosome to be present in all of them. So we do like C here as our option, right? This is an example of non-disjunction. All right, and then just kind of looking at a few more examples of chromosomal disorders. Sickle cell anemia is actually one that is being studied as a way to try to genetically edit using CRISPR to help individuals born with sickle cell anemia. It's an autosomal recessive disorder where red blood cells can sickle, and that can cause things like blood clots, or can cause it to stick and block up organs and arteries, and really painful and can be very fatal oftentimes if not treated or given medical attention. And so basically, when cells sickle, they can't transport the oxygen around the body efficiently. This is found more in higher rates in African American population in the U.S. Reasons being people with African descent, especially in areas with high amounts of malaria. We found there's a resistance to malaria having that sickle cell trait. And so just the nature of having this sickle cell trait actually has an advantage, the heterozygous one. And so it's really also a product of evolution or natural selection, right? Which we can see down here. People born with AA have normal hemoglobin red blood cells. People with the genotype AS have the sickle cell trait. Body can produce sickle cells in low oxygen environments, but immune systems eliminate them and are replaced by normal red blood cells. This is also resistant to malaria, this AS genotype. And then the SS is sickle cell anemia, so that one is severe blood clots, fatigued, and can lead to strokes or death, especially if not treated or given medical care. And then so sickle cell is more common globally in areas where malaria is more common, not just in Africa, but in parts of Eastern Asia as well. And so this is kind of going into genetics, but it's also leading us into evolution as we talk about whoever is more likely to survive and reproduce, maybe over time you'd start to see that trait show up more and more frequently in the population. And so people with the sickle cell trait have a better resistance to malaria in those areas. Otherwise, sickle cell disease would probably be kind of seen less and less in populations because there's no need to have it, or not need, need's the wrong word, but there's a disadvantage to having that allele. And only when you have some type of advantage do we start to see it come up more and more. So it's ideally for people to have that AS genotype over SS or AA in a malaria high environment. And that's basically it, right? Just chromosomes failing to separate during meiosis, specifically anaphase 1 or 2, can lead to those abnormal amount of chromosomes, which, as we mentioned, can have over 6,000 different types of chromosomal or genetic disorders. And so that can lead to a variety of different things and different problems. So that's chromosomal inheritance, and I hope that was helpful.